Time, Love, Memory by Jonathan Weiner. The book begins by talking about Seymour Benzer and his studies of mutant fruit flies. He chose to use fruit flies because they were small and reproduced very quickly. It is described that there are some mutant flies that no longer have a sense of time, which means they wake up and go to sleep at random intervals. Other female mutants show a dissatisfaction towards the male flies. Another set of mutants moves in arcs until they starve to death. Biology opened first glimpses of the foundation stones of experience: time, love, and memory. Benzer studied the connections between genes and behavior. He observed how the flies would not always gravitate towards the light. He discovers that the flies are not just creatures of exact instinct, they can make choices and change their minds. Decades prior to Seymour Benzer, Thomas Morgan, a biologist that was skeptical of Gregor Mendel's study of hereditary traits in pea plants, studied the physical mutations in fruit flies when they were injected with acid, sugar, salt, alcohol, and other mutagens. He was trying to speed up the flies' evolutionary processes. This gave Benzer a point of entry through which he could create his own experiments based on gene-behavior relationships. A book called The Machinery of the Brain was about a biologist named Roger Sperry and how he discovered you can sever the optic nerves of toads and then reattach them to the opposite sides, thus resulting in a directionally challenged toad. When a fly comes from the right, the toad would dart its tongue to the left. Humans do not have this. If you were to sever our optic nerves, you would not be able to reattach them. Sperry not only revealed this information about toads, but studied other animals as well. He found that in some animals you can sever the corpus callosum and teach each side of the brain conflicting information. Sperry wanted to find the effects of cutting the corpus callosum since the procedure was being used to prevent epileptic seizures from spreading from one side of the brain to the other. Benzer wished to observe these similar traits in fruit flies.
Benzer sent mutant flies through a countercurrent machine and found that some flies had a seizure while in the machine. He took those flies and had them reproduce and saw that their children and grandchildren had the same seizures in the countercurrent machine. So Benzer wanted to find that there are traits that are passed down genetically. So he built a fly flight tester that was about 500 milliliter glass cylinder coated in oil. And he found that strong flyers would get stuck at the top, weak flyers would get stuck at the bottom, and flies that couldn't fly at all were stuck at the end of the tube. And he dissected the flies and discovered it was due to wing muscles. Benzer needed to find a gene to prove to skeptics that these tests could be important. So he worked with flowers that opened during the day and closed at night. So he knew that they had a internal clock. But now he just needed to find a gene, and scientists Konotka found that a fly with a mutant clock gene on the far X chromosome, and he called it the period locus, in other words, the clock gene. Life comes in two ways: one, survival, and two, reproduction. The clock gene allows life to survive on a schedule, and reproduction may lead to finding a love gene. So Benzer saw that whales sing to female whales to reproduce, and the female whale will pick her mate based off of the male's rhythm. Just like how a male fly will hum a buzz to a female, and she will pick her mate based off of the male's rhythm. Benzer found a mutant fly that buzzed off rhythm, and he took his genes and transferred it into a zygote of another fly, and that fly grew up to be off rhythm too. So potentially Benzer found the love gene that controls the rhythm of song and dance.
After discovering the love and time genes, scientists sought out to find the memory gene. Memory is not something that can be measured or tested. With science, you can always wonder and assume, but not everything can be tested or proven. However, Morgan eventually found the Krebs gene. Male flies interact with the females in many ways. They have Drosophila arms that help them cling onto a female along with singing to them. This is a form of intersexual selection, which is a major component in biology. The period gene is like an internal clock, and if the flies lack this, they become off rhythm, which causes them to lose the chance to mate.
Mini evolution is the changing of a gene so that behavior can change, which then leads to the eventual change in the species. Hall and Rosbash figured out the best way to get a gene is to cut it out from one fly and to place it perfectly in the spot in another fly's embryo. Placing the gene in the perfect spot could possibly transform the fly's children's behavior. Hall and Rosbash have successfully done this so that the flies would eventually could tell the difference between daytime and nighttime, and they put the period gene in the mutant fly's embryo, giving the children flies rhythm. Hall and Rosbash cloned the period gene of a D. simulans. Then they mixed one of their DNA transformation cocktails and injected into the egg of a melanogaster. The piece of behavior jumped from one species to another. They had done mini evolution. Observers in and around biology were beginning to realize the values of flies in neurogenics. Many countries began to race and collab and map and sequence every one of the 3 billion letters in the genome across 3,600 map units. Entrepreneurs jumped on this opportunity to make a lot of money selling genes. They would generate enough different versions of enough different genes to figure out what most of the words mean, being ACTG. Period became a model for the way nested cogwheels of genes work together to produce behavior, and more fundamentally, as a model for the way genes work and for the way to figure out how these genes work. To find out what the gene sequence meant, had to alter the spelling on a letter at a time, G to A or A to C.
Along with many animals and insects, humans are instinctively social animals, although some scientists argued that humans were past that. This led to a discussion over nature versus nurture in relation to our tendencies and behaviors. While criticized for his work, Benzer continued searching for answers, primarily with his flies. He and his second wife studied the effects of antibodies on the flies and soon moved on to the human central nervous system. The antibodies revealed many molecular and mechanical consistencies between fly and human brains, and soon the universal family likeness of genes was confirmed. Scientists noticed that gene periods had distinctive runs of letters that would repeat many times and produce the period protein. This could be seen in flies collected in many experiments. Those experiments found that the frequency of repeats varied by the individual, and the farther the repeats were from the center of the gene, the longer their runs would be. This is true in humans too. In fact, research in this area led to the development of the design for human DNA fingerprinting technology used today. It was hypothesized that these repeats might help us better understand genes such as the clock or time gene. The clock gene was discovered in mice, and soon it was found on the 66th region of a fly's third chromosome. The author points out that we must think of genes not as sealed envelopes that we receive from the previous generation before us and pass on unopened to the next, but as messages that we read and reread continuously for the rest of our lives. Many animals, like flies, snails, and dogs in experiments, learned from experiences that were painful. This was evident in many experiments that set out to prove conditioning and behavior modification was possible. Although the learning processes of the subjects in these experiments took several weeks and even months, they soon learned to react to certain conditions and experiences that hurt them previously. The CREB gene played a role in this, as it is considered to be a long-term memory gene.
How does this book relate to Bio 100? So Benzer and Morgan are both biologists, and Benzer is the one who connected classical genetics and molecular biology by watching two chromosomes line up and exchange genetics. This led to GMO foods, which is a genetically modified organism. And that leads into mini evolution because it changes the gene, which potentially changes the neuron, which potentially changes the brain, and that changes behavior. Also, Benzer studied how bacteria reproduces by binary fission, which is an asexual process that means that the reproductive bacteria are identical or cloned. Also, Benzer talks about how the structure of the DNA was found by Morgan, which is T, A, G, and C.